Okay guys, so this video is about the lab utilities. So in this video we will tell you about what are the basic components that you should know about, what are there in a lab, and you should be aware of them. So even if you don't know in detail, it's okay, because we can provide you some information on that, but I just want to introduce to you what they are and how they look like, so that at least when we talk about them in our different video lectures, you feel that you know them just by term. So the thing that you see in front of you is we call it a measuring cylinder. So as you can see, it's of 100 ml measuring cylinder, and then here you can see, so 100, 90, 80, 70. So that means you can measure a volume of cylinder can be of different volume range. So let's say here these are just a measuring cylinder, and you see the aluminum foil on the top of them, they're covered, they're sterile, they're not contaminated from inside. Yeah, so this is a measuring cylinder here, and the general operation about them is very easy. So we'll take example of our 100 ml measuring cylinder, and we will measure some volume. So right now I have some water in my hand, and I will show you in a very general way how we measure it. So you can see it here. Now it's filling it up. Let's say you just want to measure only 90 ml. So you fill it like this. Then you can look at here. So this is 80, here is 90, but we only get about the right feeling when you put it in a plane. So now we put it in our bench, and then we can see. So this is 90 here, and it is filled till here. So if you just shake a little bit, you can see it. Yeah. So that's how you measure, and then you add it whatever you want to add. So we will tell you about in our buffer exercise where we use the measuring cylinder. Okay, cool. I will now introduce you with another set of lab utilities. We call them beaker, and I hope you already familiar with the term beaker, because if any of you have attended the chemistry classes, you have heard about the beakers, right? So here you can see the three types of beaker. They are of a different volume. Let's start with this one. So here this is a plastic beaker, and here you can see it's a 250 ml. So you can also use the beaker to measure the volume. Let's say this one is 2 liter, 2000 ml, that's 2 liter, and then this one is 500 ml. So these can also be used to measure the volume, but they are less accurate than the measuring cylinder, and again they only give you the rough estimation of volume. So beaker can also be used. They can be used to measure a large amount of volume. For example, you can see here in 2 liter beaker. So if you want to prepare a media 2 liter, then you can use this much big beaker. Oh, it's okay. So I'm sorry. The media is like, if you want to put the bacteria, you have to prepare the culture media. So we can prepare those culture media in these beakers. Yeah, great. And then I will introduce you with the next set of lab utilities. So now you see in front of you conical flask, and if you know them, that's great. If you don't know them, I will tell you. So these are the conical flasks. You can see them. They are a conical shape, and we use these conical flasks to grow the cells, to do bacterial culture, to yeast culture, sometimes animal cell culture, and sometimes tissue culture also. Yeah, so they're very important. And yeah, so they're also available in different volume, and you can see it here. So the highest volume, this one, will show you the 50 ml, and we will only use this to grow 10 ml of culture, not more than that. So there should be some relation. So if the volume is let's say 2 liter, you should not grow them more than 50 ml of culture in the 2 liter, or more than 100 ml of culture in there. Yeah, I mean, it depends on different type of culture. We will talk about them detail in our microbiological tutorial, but here it's just like to introduce you what are the conical flasks and how they look like. So you can see here, this is I hope it will be visible. Yeah, so this is 250 ml. We use it to grow 50 ml of culture, and then you can see again here. So yeah, it's 1 liter, and we grow them not more than 500 ml of culture in this. Yeah, great. So this is the third set of utility that you should know. Okay, so now these are the fourth category. We call them Scott flask, Scott bottles actually. Yeah, not flask, Scott bottles, and they are made up of very strong glass. Yeah, so they're very good quality glass, and you can see them. They're very transparent and very good quality. Yeah, and in this also you can see that there is some kind of volumetric ratio, the volumetric estimation. So it is like 400 ml till here, and this is a 500 ml flask. So you can find it, it's written here, it's a 500 ml Scott Duron bottle. And the same is, so this one is 1 liter, this one is 100 ml, and this is 50 ml. Now they are very important part of lab utilities. We prepare all the buffers in these bottles, we prepare all the chemicals, we also we use them to prepare the sterile water, sometimes to prepare media and etc. So they are a very important part of the lab, and as you can see them, they are closed from the top and they're very tight packing. So we use them to store things, so as to store buffer, to store water, to store anything. Yeah, you got it right. Okay, so now I will introduce you to two different things. Here you can see two stands. We call them falcon stands, and we call these tubes falcon tubes. As again you can see, there's written 50 ml. Yes, now you're going to get it. So this is 50 ml, and you can see them, there are 15 ml. So this is 15, this is 50, and these stands, we call those stands of falcon stand, and we use these tubes for a very important purpose. So for example, if you want to prepare something 20 ml, let's say you want to prepare 20 ml of magnesium chloride solution, and you can use these tubes. And the good thing about these tubes, they are also quite accurate. So if you see 20 ml here, that means it's actually 20 ml. Some people use them to do cell culture. Some people use them to use a special type of buffer. For example, if you want to prepare anything in a small quantity, you can actually use those tubes. Yeah. At that time, you don't prefer the 50 ml or 100 ml Scott bottles. You rather like to have more handy these plastic tubes, so you use them and you throw them. Yeah. And it's just a short reminder. So the rest of the thing that I showed you before that, you cannot use and throw them. So that you use them, you wash them, you sterile them, you make them sterile using autoclave, then you reuse them. Here in these tubes, generally depends on the lab. So some labs prefer to reusing them. Yeah, they are autoclavable. But in my lab and most of the people, they don't prefer to reuse them, so they use and throw things. Great. Okay, so then probably this could be very important lab utilities. This you can see here, this thing is Eppendorf Stand. We call them Eppendorf Stand. So this is just like a normal stand. You put them here, and then in these holes you can put the Eppendorfs, and how we will see this. So then now you can see these bottles, these are the Eppendorf tubes. We use them in a different purpose and variety of purpose, and they can be of different volume. Generally the most common and most popular Eppendorfs are of 1.5 ml. And why they are closed with this, with aluminum foil bottles, because we use them after sterilization. So we sterilize them using autoclave. For autoclave, I will tell you in detail. They are here. This tutorial is just to make you familiar with the different terms that are common and you should know them. Yeah. And then yeah, exactly, you see this. So now I can pick some tubes for you and I can show you what they are. So this is a 1.5 ml tube, and there's somewhere written 1.5 ml. Let's find it out where it is. Just give me a second. So you can see this is 0.5, this is 1, and this is written 1.5 ml. So this is 1.5 ml Eppendorf tube. That's how we do it. We close it, and we can open them like this. Great. And this is the stand which we use it. For example, put it like this so you can see it. You can see it from the top. So that's how you do. You put them, just ignore those small holes. They're for the different Eppendorf tubes. So you put them like this. And on the top or on the side, you can use it to write the name, something if you want to specify something. So this is the most important lab utility. We always use the Eppendorf and Eppendorf Stand. I mean, we do most of the reaction, most of the test in Eppendorfs. So you should know that one. Great. So far these are the common lab utilities which I think you should know. Yeah, in front of them you see some more stuff, and we will talk about them in a while.